Welcome to our lab podcast on the circulatory system. Now, as we begin to look at different blood vessels and different sizes, what we've taken here is we have taken a piece of mesentery from likely the abdominal cavity, and you can see tons of blood vessels coursing through this very thin membrane. And this gives us an excellent view of these blood vessels unobstructed, so we can see the whole thing and really the three-dimensional detail of these vessels. So as we look a little bit higher magnification, in many instances around the dark blood vessels we can see circular rings of objects, and these are going to be adipocytes. But then as we go in higher and higher magnification, we can see structures that look like this. Certainly you can make out just the faint edges of adipocytes that are out of the plane of section, but here we have a small blood vessel, and in all likelihood, we can't really see the red blood cells inside, but when you see these nuclei running almost around in this direction, you can pretty much bet we're looking at the nuclei of smooth muscle cells coursing around, or possibly pericytes. It's again difficult to tell with it so transparent. And then these nuclei that are running longitudinally along, these are going to be the endothelial cell nuclei. Here again we can see a nice colored image. It's fairly easy to pick out the adipocytes, unilocular adipocytes, and we can see different sizes of these blood vessels. We can't make out any other histological structures and features, but you can clearly see the red blood cells in single file fashion. So this is either going to be a capillary, or more likely a capillary, or possibly a venule. We would need to look at the histology of those vessels to be able to determine that. All right, now let's begin to look at these different sized vessels and their different features and the different layers that are present that we learned in lecture, as we look at these in higher and higher magnification and greater detail. So in this particular illustration, we can see a muscular artery, we can see a fairly large vein, we can see some smaller veins, and then we're going to get down to the point that we'll be looking at arterioles and capillaries. Now once again, as we begin to look at a vessel, we're going to recognize the lumen of these vessels, and we have two here. We're going to recognize the tunica intima, which is the one opposing the lumen. We have our endothelium. The middle layer is the tunica media, and then it's fairly easy to recognize the tunica adventitia. Now, within the media of this vessel, we can see a number of these circularly arranged nuclei. So this is going to be the smooth muscle layer. And this relatively thick muscle layer lets us know that this is a muscular artery. Over here, again, we can't see the entirety of this vessel. This is an inset from the previous illustration. So from that, we know we have an extremely large lumen. We do have some smooth muscle, so we are looking at a vein. Now here we have a different section through the tissue, and these are the smaller vessels, and we know they're smaller and the luminal diameter is different because it's easy to look at the nuclei. Here we can see the nuclei. There are not nearly as many of these in this one as we saw in the previous image, so this one is much smaller, as is the diameter of this vessel. But again, we're looking at the diameter of the lumen versus the thickness of the wall of the vessel. Again, we can see intima, the muscular layer of the tunica media, adventitia on the outside. Here we have much larger lumen than we do thickness of the wall and adventitious. So this is still going to be a vein, and this is still going to be a muscular artery, because even with this we can count upwards of 6 to 10 layers of smooth muscle. Now, looking at a different plane of tissue, this is a skeletal muscle section. We can see our sort of marbled muscle out here, and then we have some fairly large vessels that we're going to look at the walls of these. So in this section, lumen on this opposing side, and from the previous illustration, we know that these are fairly large luminal diameters, but here we have a fairly thick muscular layer. Here we have just a couple of layers of smooth muscle before we get to the adventitious. So here we're going to have a muscular artery, and here we're going to have a vein. Looking a little bit closer, again, we can fairly easily identify the endothelium. So if I ask you to identify these cells, you would say endothelium. See, we're moving away from the classification of simple squamous epithelium. Now we're going to identify them. Here we have subendothelial compartment, and that's still, remember, part of our tunica intima. And then we get the tunica media, smooth muscle cells. We've got some elastic lamina running through here, but predominantly we see the smooth muscle. And then we have our tunica adventitia. Notice in this, we do have blood vessels in the adventitia, so do you recall what we term collectively these blood vessels in the adventitia of blood vessels? It's called vasa vasorum. And then we also have a little nerve bundle right here, also present in our tunica adventitia. Now, as we went through in lecture, we talked about the lymphatic vessels and veins having unidirectional valves built into them, almost like the cusps that we see of our semilunar valves. Well, these are sections through some of those leaflets of the valves in veins. All right, just a few more examples, and once again, this is what you have to do as you look at histological slides. You have to find as many different examples of muscular arteries, veins, elastic arteries, arterioles, capillaries, as many as you can, so you get comfortable with the structures and the associations of where one is versus another. The color's going to be different. The stains can be different. The shapes are going to be different. So you have to be able to use differential diagnoses, much like you will in medical school, to identify and classify these different structures that we're now moving into with our organ histology. So we're going to look at one of the insets that we saw, much higher magnification. So once again, we are looking at a blood vessel. We don't know what it is yet. And this stain is unusual than is from what you've seen in the past. The blue is going to be staining largely our elastic fibers. And that's what's going to give it this look in here, as well as mixing in between the red nuclear stain that we see. This is going to be our tunica media, and these nuclei are going to be of our smooth muscle cells. So we see we have a relatively thick tunica media. Here we have our tunica intima with our endothelium. And then what do you think this pink line represents? That's the boundary between the tunica intima and the tunica media. That's going to be the internal elastic lamina. And then as we get down here, really hard pressed to see, but it would be in this general location, the external elastic lamina. And that's going to be the demarcating line between the tunica media and the tunica adventitia. So we're looking at a muscular, fairly large, thick muscular artery. Now this is an interesting section through a blood vessel. What we are sectioning through, on the right we have a longitudinal section, and we can see we've cut the nuclei of our smooth muscle in cross section, somewhat getting some of the endothelial nuclei in here, especially at this location. These are red blood cells. But then the blood vessel bends, and we're sort of getting a tangential cut through the circularly arranged nuclei of smooth muscle before we lose the plane of section and that blood vessel goes out of the plane of section. But even in this case, we can see the thickness of the media. We can count the number of cells that we have here. Again, maybe 6 or 7 layers in some places. So we're still looking at a muscular artery. All right, different stain, looking at blood vessels. Follow this inset as we get higher and higher. This is going to be a low magnification view, as you can see, of an elastic artery. So we're going to expect to see a number of layers, intermixed layers of smooth muscle elastic lamina, smooth muscle elastic lamina. And as we look at the inset, that's exactly what we see. This almost looks like a negative image because all of the clear white areas that we see are in fact elastic lamina. And I like to compare elastic arteries to lasagna because these look like the ruffled edges of lasagna noodles with the meat sort of intermixed. So these are the smooth muscle nuclei intermixed with elastic lamina. So this is our tunica media, adventitia here, tunica intima on the surface of an elastic artery. Here we just have a higher magnification look at the clear or the light colored elastic lamina and the dark blue of the smooth muscle nuclei. Okay, continuing to look at vessels. I believe this is going to be another elastic artery. This one's stained almost predominantly for the elastic fibers, and in this case, we have a number. It's almost difficult to see the smooth muscle cells, but there are smooth muscle cells in here as well. So this is the tunica media, intima. We have the tunica adventitia, and this is an interesting section through a blood vessel, and it's just a very long longitudinal section, and we can see the media, the tunica media, the smooth muscle cells and their nuclei cut in cross section, and the tunica adventitia, and then the dark line of our tunica intima, predominantly our endothelium. Another type of stain, again, this is more of a nickel stain, and that's why we have this yellow brownish color. And again, it's really going to highlight the elastic lamina and elastic fibers that we have, not just in the vessel, but you can see a lot of the dark fibers in our connective tissue, adventitia of the vessel, and intermixed between. So this really exquisitely highlights where we have the internal elastic lamina and our external elastic lamina, and then we have the discontinuous elastic fibers in our tunica adventitia. Once again, a lot of elastic fibers present here. The yellow is going to be smooth muscle cells, and then we can see again elastic fibers present in our tunica adventitia. Again, using this stain to look at the different layers, and it's difficult to see, so we're going to look at the inset here. And here we have the inset. Once again, we have lumen, tunica media, and then we have relatively thick tunica adventitia. And remember, for our veins, that's where we're typically going to see the thickest layer being the adventitia. So even just looking at this, we want to suspect that we're looking at the wall of a vein. All right, getting back to our H&E stain that we're fairly accustomed to seeing by now, we're going to look at various insets of this large vessel. And again, whenever we're looking at the folded lumen, almost collapsed lumen, that's sort of my first indication that we're looking at a vein. So we'll take that inset and look at higher magnification. Once again, we can see the lumen near the top, the intima, a few smooth muscle cells, and then mostly we're looking at our adventitia layer. Now, we do have some vasa vasorum down here, but look at the thickness of the adventitia layer. Incomplete smooth muscle, and you're not often going to see these big bundles cut like this. So most of the time you're just going to see your relatively dense, irregular, or maybe even loose type of connective tissue in the adventitia of a vein. Higher magnification, again, intima on the right, little bit of tunica media with our smooth muscle, and then we get into our adventitia. Again, this is not a real common occurrence, seeing these large smooth muscle bundles here. Again, cut in longitudinal section. These are circularly arranged, and these are longitudinal bundles. You won't often see that. All right, again, looking at different layers. Let's follow this inset. Again, we have our endothelium on the right, tunica intima, which would include the subendothelial layer, and then a lot of smooth muscle present here. Now, this is difficult because we didn't really see how big this lumen was. So it's difficult for us to really classify this vessel just on the images that we've seen so far. All right, so now we're starting to get smaller even. We can see the bulging nuclei that are present there. This is going to be our endothelium. We've got our tunica media, and in this case, maybe 2 or 3 layers. So we're getting down to the point that we're beginning to see arterioles. Now, usually you will have partners. You will have an arterial partner and a venous partner. So if this is an arteriole, then I am somewhat suspect that we're looking at a venule in this case. And see, they say this small muscular artery 3 to 4, that's that boundary between muscular artery and an arteriole. I'm going to go ahead and call this one an arteriole because you're really splitting hairs at that point. And again, we didn't see the lumen of this completely, but if this is an arteriole, I'm probably going to call this 99% of the time, call that a venule. These are pretty cool images that we see here. We're seeing somewhat of a tangential cut through a blood vessel. We can see our red blood cells, but we see a lot of white blood cells, and they are actually neutrophils, polymorphonuclear leukocytes. We've got our endothelium here, possibly a pericyte, but again, when you look at that luminal diameter, we're probably looking at a venule. Here we have endothelium, and we have 1 cell around. So we are really getting close. I mean, this would be a very small arteriole, but then we absolutely have capillaries here. 1 endothelial cell, the diameter is that of the red blood cell. Once again, we have this vessel, our endothelium. Okay, I don't know what's doing that. Okay, this is making me mad. Here we have another vessel. We can see our endothelial layer. We've got essentially 1 layer of smooth muscle around. Can't really see the diameter. We don't have a red blood cell in here to measure. But with that 1 smooth muscle layer, then probably going to call this an arteriole. Out here, these are either going to be venules. If we had some blood cells in these to know whether these are only white or some red blood cells, then we'd be able to tell if these are lymphatic vessels or, in fact, venules. At this point, associated with an arteriole, I'm going to go with venule. Once again, here we have some small vessels. We have an arteriole, got that one smooth muscle cell around, endothelium on the inside, possibly a venule that is collapsed here. But in this case, we see white blood cells and no red blood cells in this fairly elongated, only endothelium, so this is going to be our lymphatic vessel. Now, shifting gears, moving into our EM images, now we're going to be looking at these fine structures as we get to these very, very small luminal diameters. And once again, we can see our endothelium on the inside. We have these occluding type junctions between our endothelial cells, one cell on the outside, and possibly some dense body material here. And so here we can see the cytoplasm also around this side. I'm gonna call this an arteriole because I believe this is a smooth muscle cell. Here we can see the caveolae, or the pinocytotic vesicles adjacent to the lumen. These are the pinocytotic vesicles of our endothelium, and these are the caveolae of our smooth muscle cell. Remember, these are the structures that store calcium in smooth muscle, and these are just the transport vessels for our endothelial cells. Now, I don't like using that term caveolae for endothelium. I would rather use vesicles, pinocytotic vesicles, just to make sure we distinguish what's happening in an endothelial cell versus what's happening in a smooth muscle cell. Again, just more images of that same field that we were looking at. A little bit lower magnification. We can see collagen fibers and cross-section, longitudinal section near the bottom. There's our nuclei, possibly a little nerve bundle up at the top, unmyelinated nerve bundle. All right, looking at some more images here, you can actually see a myelinated nerve there on the right. But as we look more closely at the inset there in the middle, we're going to see some tight junctional complexes. We can see several layers here, so this is going to be smooth muscle layers, so we're still going to call these arterioles at this point, with some nerve bundles out in the adventitia. And again, here we can see those occludens. Again, here we see the occludens type junctions present between these endothelial cells. Now, we've seen this image in lecture. This is our sinusoidal type of endothelium. This is present in the liver. Here we have 2 liver cells, tons of mitochondria, some rough endoplasmic reticulum here, nucleus in this point. And then here we have our discontinuous endothelium. Looks like it's just shredded, but remember there's such large holes present in these membranes that it's going to give us this appearance in a cross section. So this is our sinusoidal endothelium. Once again, another look, portions of our endothelium here. And we're actually going to have microvilli from these hepatocytes. We'll talk about that when we get to digestive system, that are projecting into this space beneath the sinusoidal endothelium. But right now we're just focusing on those endothelial cells. Here we are in the kidney, the glomerulus of a kidney, and this is where we have our fenestrated endothelium. You might remember this from phys class. These are the pedicels of our podocytes. On the outside, there's our basement membrane. You can see the 3 layers even in this little mag, the 2 lamina lucidas and the lamina densa in between, And then here we have the fenestrated endothelium of our glomerular capillaries. So a little higher magnification. Again, we can have the openings here, the pores, the fenestrated pores, but these are going to have these little membranes across them. Can't really see them so well in this particular image. And then here are the pedicels of our podocytes. Here's a podocyte process, and there's a pedicel attaching down to the basement membrane. There we can actually see a slit, a membrane slit between those pedicle slits. So you have those membranes there on the other side as well. We don't call them fenestrations on that side, though. And then here we have our continuous capillary. We can see our endothelial cell, one endothelial cell thick, possibly pericyte cytoplasm here. And then we have our occludin types junctions, fascia occludens here between these processes.